Okay, go ahead and get started with our next lecture topic on Mendelian and quantitative genetics. So, a rather crucial aspect to biology, a lot of terminology, so please pause throughout this recording to make sure you're taking the proper notes and understanding everything as we go. And we will make sure we talk about everything in detail together when we meet next. So, Mendelian genetics, what does that even mean? Well, so modern genetics began when the Austrian monk Gregor Mendel documented the mechanism of inheritance. So, this guy basically was an Austrian that went to a monastery and was studying science and math and was noticing variability within pea plant populations. And so he basically was able to look at combining different types of peas together to determine what the offspring of those peas were going to produce. He was able to publish a lot of his findings in 1865 before he died about 20 years later, but the significance of this work was not even appreciated really until the year 1900 by a few more researchers, which built upon these ideals. So, pea plants typically self-fertilize. So within the pea plant itself, it will have the female and the male parts. Mendel specifically took pollen or male parts from one plant and put it with a female part of another plant with maybe a different characteristic, such as moving white and purple flowers together with female and male parts from one or the other, to see what types of traits might arise from this, whether they would all be purple or all be white or whatever he was looking at. And so he was able to determine what was going on with these different plants and how that might be influencing their offspring. And so to understand a lot of this stuff, we gotta take a step back and make sure we understand some terminology that is representing this material. So chromosomes, which we should be already familiar with, are analogous to the pages in the instruction manual. So each page is going to contain thousands of words. Different types of cells use different words in different orders. Well, genes are segments of DNA that code for proteins, which we should also be familiar with. These genes are analogous to words in an instruction manual for building a human. And so specific aspects of muscles are going to have specific genes that are encoding things for muscle cells that they need, such as higher amounts of mitochondria, which we learned on the last exams material, or things for eyes, such as eye color. So, a few more terms. We have genotypes and phenotypes, which you're probably familiar with from high school biology, but we'll make sure we rehash it out here. So, a genotype is the specific part on the DNA sequence, or in the gene, that is going to be coding for a trait. So it's a combination of alleles. So homozygous is going to be the two same alleles within the genotype, whereas heterozygous is going to be two different alleles. We'll come back to this idea throughout. So homozygous is the same allele represented twice, and the heterozygous is there are two types of alleles within that genotype. Phenotype is the physical outcome of the genotype. So it's a product of the nature of these alleles. It's what we physically can see or feel or hear, such as rough skin, blue eyes, someone's deeper or higher voice, someone's really nice sounding voice for vocals, whatever you can actually tangibly look at or see or feel, right? We're not looking at DNA, so that's why it's called the genotype. When it comes to different types of alleles, we can call it either dominant or recessive. And so a dominant allele can mask a recessive allele. So if a dominant allele is present, it will be expressed. A recessive allele can be masked by a dominant allele. So in the absence of a dominant allele, the recessive allele will be expressed. But if that dominant allele is there, it will be masked by the dominant allele. Incomplete dominance is when we kind of get a mix between the two types of alleles. So say red flowers are dominant and white flowers are recessive, but then we mix those two. So we mix a white flower with a red flower, and then we might get a pink flower. Well, that's a mix between the two. So this is an example of incomplete dominance. Codominance is when both alleles are fully expressed. So codominance can be something like sickle cell anemia, which we'll get to shortly, where you might be a carrier for a trait, but not necessarily express that with what we can actually see, but you could possibly hand that trait down. So both are being expressed, we just can't necessarily see it. And this is an example of codominance. So when it comes to dominant alleles, we represent these with capital letters. And so you can see the left chromosome here, or I guess the left chromatid, right? This would be a chromosome together, and it's a pair of sister chromatid. The left one would be all capitals, and the right one is going to have two recessive or lowercase ones and one capital. So this chromosome or this chromatid has all dominant alleles, which will mask all recessive alleles present on this one that match up with that letter coordinate. So we could say large T would be for tall genes and little t for short genes. This is arbitrary. We're just kind of saying this for the sake of understanding that dominant alleles and recessive alleles can code for different phenotypes. And so we could say that the A allele that is capital is dominant over the lowercase recessive A allele. Punnett squares, which you probably are familiar with from high school, are going to help us determine what type of offspring will come about based off of genotypes that we're mixing together from the parents. So this will use gametes from the parents to predict possible phenotypes of the offspring. A parent that is heterozygous for a trait, heterozygous meaning two different ones, we have a capital A and a little a, can produce exactly that, two possible gametes, either a big A or a little a. A parent that's homozygous for a trait, big B, can only produce gametes with a big B. They don't have a little b, so they're the same, homo being same. So they have two of the same alleles, two of the different alleles. And so this would be one parent here having, so in this scenario, we could look at this homozygous trait having two big B's for this particular parent. And then this particular parent is also heterozygous for a different trait with a capital A and a little a. So when it comes to the genetic disease cystic fibrosis, we're looking at a recessive condition. Individuals with cystic fibrosis are going to have two copies of the cystic fibrosis allele, which are both recessive. This will produce a defect in the chloride ion transport, causing lung infections and to shorten the lifespan. Carriers of cystic fibrosis can have one cystic fibrosis allele, but not the other one. So they're not exactly expressing cystic fibrosis, but they can still pass that along to their children or their offspring. And we can kind of see that represented within this particular Punnett square. So the offspring can be either homozygous or heterozygous. It all depends on the parents and the possible gametes that come along. So when we talked about meiosis in the last exam material, we talked about sex cell production, and those sex cells have half the genetic material. So they wouldn't have two sister chromatid, they would have one of them. So on one of those sister chromatid, they would either have a big F or a little f, right? So here's the eggs, big F, little f. So these Punnett squares can be used to help predict what the combinations of these different sex cells or gametes can be to produce offspring. And so we have the sperm from the dad over on the left and the eggs from the mom up on top. We have both alleles represented in both populations. So we have a big F crossed with a big F, giving us a homozygous dominant large F. All right, and this particular color is going to be 25% of the four possible options, right? 1 in 4 chance. So we can go over here and look at our little chart. 25% chance that this child will not have cystic fibrosis. They won't carry it, nor will they have it. Well, we can cross that big F with a little f, and we'll get both alleles represented. The fact that we have a dominant trait in this large F means that this person will not have cystic fibrosis, but the recessive allele being present means that it is within their genetics and they can pass it on to their offspring. And so we're looking at a 1 in 2 chance, or a 2 in 4, I guess, 2 in 4 chance, or 50% chance that this child will be an unaffected carrier of the cystic fibrosis allele. Unaffected means that they do not have it themselves, but they're a carrier of it, so they can give it to their next offspring. And then we have this little guy down here, which is the exact same odds as homozygous dominant. We have little f and little f coming together for a homozygous recessive, and since there's no dominant trait masking the little one, both of these will be expressed, and so this is a 25% chance that this child will have cystic fibrosis. And so it's a very unfortunate circumstance, but two parents that are heterozygous for cystic fibrosis have a 1 in 4 shot of having a child that will have cystic fibrosis, even if neither of them personally have to deal with that disease, but they could have a child that could get it. Genetic diseases continued. Next, we have Huntington's disease. So Huntington's disease is a dominant condition, unlike our recessive condition in cystic fibrosis. So this is only one dominant allele needs to be present, and this offspring will have Huntington's disease, which is a fatal condition, producing an abnormal protein that clumps in nuclei of cells. And this is a serious issue when we have nerve cells that don't mitotically divide regularly, and it leads to a horrible degradation of brain tissue. So with Huntington's disease being represented in our cross here, our Punnett Square cross, we have mom is completely recessive. She doesn't carry it whatsoever for Huntington's. No Huntington's genes in mom's pool. But dad carries it. He actually is going to get it by the end of his life because he has the big H, right? So this is dominant over the little h. So looking at our cross here, there's a 1 in 2 chance, or 2 in 4 chance, 50% chance that the child's going to get Huntington's disease because of this dominant allele. And then the exact opposite is 50% chance that this child will be unaffected and not carry the allele for Huntington's disease. So this is a dominant condition. So far we've had a recessive condition in cystic fibrosis, and now we've had a dominant condition in Huntington's.